Thank you, Melanie. It's a pleasure to be here, and it's a pleasure to follow Dr. Lamerle's talk. Actually sets up mine well. And I'm going to talk about this topic in general initially, and then segue into a specific study that we're funded to do. We have one of the EPA and IEHS Children's Environmental Health Centers that I know several of you either participate in or know of. And our particular center is the Berkeley-Stanford Children's Environmental Health Center. And the study that funds is called CHAPS, or the Children's Health and Air Pollution Study in the San Joaquin Valley. So as you've heard from Dr. Lamerle, there's an epidemic of diabetes in the U.S. I don't think this is news to anybody in the audience. A lot of Americans are diabetic. Diabetes in children is usually, or has been, thought of as mostly being type 1, which is an autoimmune disease where pancreatic beta cells are actually damaged by inflammation. And so there's decreased insulin that's secreted by the pancreas into the blood. But the rate of type 2 diabetes in kids is increasing, especially among minority populations. And, these are similar data from the CDC that Dr. Lamerle showed. The epidemic of obesity, as she also talked about, is incredible, that 34% of us are overweight and another 34% are obese. So that means there's only a third of us that are in the normal weight range. And the percentage of children who are obese is also increasing like crazy. And I don't know how that shows, but again, similar slide from CDC data to what Dr. Lamerle talked about. So we have, a huge problem, or I guess I say huge problem these days. Finally, somebody got it. Both Bernie and Trump say that, right? So the obesity epidemic is thought to be primarily due to, increased caloric intake, high consumption of high fructose containing drinks and high caloric density fast food, and decreased energy expenditure from a sedentary lifestyle. So more calories in, less calories burned. But as Dr. Lamerle already pointed out, it's more complex than that, sedentary lifestyle. Animal evidence suggests, and she showed some of that chemicals in the environment may be obesogens. And I really liked how she showed that calories in and the sedentary lifestyle really couldn't explain the obesity we're seeing in animals. I actually have learned that today, and it's important information. So in addition to Firemaster, organochlorines have been associated with obesity, and bisphenol A, misspelled there. But there are other environmental exposures as well. So there's dietary composition, and I've already alluded to it. The kids that are most at risk for obesity based on social factors, structural determinants of health, also tend to have the worst diet in terms of caloric composition because of lack of good food access in urban centers, for example. The gut microbiome, somebody asked Dr. Lamerle about that. I'm not proposing to get into this, but I think it's an important issue with regard to both the obesity and diabetes epidemics, since the gut microbiome plays an important role in immune system development and function, and also may have direct effects with regard to fat absorption, caloric absorption from the gut. The built environment, is important with regard to exercise and food consumption. I've already alluded to that. I've alluded to the food side, but exercise side, again, minority kids of low socioeconomic status who live in urban centers probably don't have much green space to exercise in. Probably the funding for after-school basketball programs isn't there anymore, and probably are afraid to go outside and exercise because of crime in their neighborhood, not to mention other things. So I'm going to focus on another area where there aren't a lot of data, but some, suggesting that ambient air pollution exposure may play a role in both obesity and diabetes, and particularly traffic related air pollution. And then probably don't have to convince this crowd that we think that in utero exposures to air pollution may play a role because of the Barker hypothesis with regard to fetal programming for subsequent disease in the life course. And maternal exposure to air pollutants definitely can affect the fetus. So linkage of diabetes and obesity. Type 2 diabetes was thought of as an adult onset disorder. It used to be called adult onset diabetes. And it's a disorder of glucose metabolism, and it's classically defined by insulin resistance, as Dr. Lamerle already alluded to, where the body's cells fail to take up glucose from the blood. And we're showing a normal cell with glucose and insulin receptors, and an insulin-resistant cell where this is showing downregulation of actual cell surface receptors. It's one possible mechanism of insulin resistance. 80% of those who develop type 2 diabetes are obese, whether they be adults or kids. And classically, obesity, as I said, is associated with insulin resistance. But there's increased evidence that it's also associated with beta cell failure. The beta cells are the cells in the pancreas that secrete insulin. As I mentioned in my comment after Dr. Lamerle's talk, both diabetes and obesity are associated with increased systemic inflammation. And there is this this concept of adipose tissue pancreas axis, where there's sort of crosstalk. Dr. Lamerle actually mentioned this as well. There's crosstalk with regard to hormonal effects, but there also may be crosstalk with regard to inflammatory effects. For example, adipose tissue has been shown to have infiltration by inflammatory cells such as macrophages that have been shown to increase secretion of tissue necrosis factor alpha, TNF-alpha, an important inflammatory cytokine, which has also been shown to have a direct effect on beta cells. So TNF-alpha secretion from inflamed adipose tissue, activated adipose tissue may cause beta cell failure in the pancreas. So how about air pollution and obesity? So there are several studies. There are some studies in adults, which I'm not going to go into, but several studies, not too many, with regard to kids. And my former colleague, Michael Jarrett at Berkeley, who unfortunately we lost to UCLA, found that higher levels of traffic were associated with higher attained BMI in children aged 10 and 18. This was an LA-based study. Mike worked with the Children's Health Study team, and specifically Rob McConnell. That Children's Health Study is the most important air pollution study that's ever been conducted with regard to children, in that they were able to follow a large number of, thousands of kids from age 9 through age 18, primarily focused on lung function initially, but they were able to look at other things as well. And, this was a crude marker of exposure that they used. It was traffic density within a buffer, several buffers, 150 and I think 500 meters from the residential home. They went on to try to get a little bit better exposure estimates to traffic-related air pollution. They used NO2, nitrogen dioxide, as the The surrogate exposure for traffic, it's a pretty good one, and they were able to estimate NO2 concentrations at the home. It's probably a little bit better of an exposure assessment approach than traffic density in a buffer. And they showed that that was associated with BMI. And then one of the Columbia Children's Environmental Center investigators, Dr. Rundle, found that early life exposure to polycyclic aromatic hydrocarbons, which are created whenever carbon-based compound is burned. Hey, Gina. pHs are generated when you burn gasoline, diesel, wood, tobacco, and when we grill our food. But pHs are complex hydrocarbons that have a lot of toxicity. They generate reactive oxygen species, which can lead to injury and inflammation of cells, cytotoxicity. Some of them are carcinogenic. And this Columbia study found that early life exposure to pHs was associated with increased BMI and obesity at age 7. And then a study from Mexico City showed that kids exposed to high concentrations of ambient fine particles, PM2.5, had high blood leptin levels, that adipokine hormone that Dr. Lamerle talked about, which those high blood leptin levels are associated with increased risk of obesity. I mean, they're actually associated with obesity. So here are some of the data. This is the first Jarrett study using the traffic density metric with a buffer around the home of the child at 150 meters on the right, and then 300 meter buffer, 50 on the left, and 300 on the right. And you can see that there were statistically significant effects of the traffic exposure at 150 meters and almost significant at the 300 meter buffer. And this is their growth data where they actually have modeled nitrogen dioxide concentrations at the home. And their model was pretty good because they took a subset of the Children's Health Study kids and actually measured NO2 at homes, and then were able to model from distance from freeways and roads, the NO2 concentrations for all of the cohort. And this is showing baseline exposure away from, with low NO2, I think it's the lowest 10% of the cohort versus at the other end, the 90th percentile. And there was definitely a exposure response here. This is growth of BMI. And this is the Rundle PAH data. And BMI less than 5 years of age on your left, and at 7 years on your right. So that's air pollution and diabetes, and obesity in kids. Air pollution and diabetes, there's even less data. Several studies, including 2 by Michael Jarrett, have shown associations between diabetes in adults and exposure to traffic-related air pollution. He's got a new one coming out that's going to show an association between ozone and diabetes in adults. A few studies in kids. 2 studies have shown associations between ozone and type 1 diabetes, which is interesting because that's primarily an autoimmune disease. Ozone does have effects on the immune system, and at least one of those studies are also an NO2 effect. Even though ozone is generated from traffic emissions, it's not usually thought of as a traffic-related air pollutant because it's regionally dispersed, whereas traffic-related air pollutants are usually common parlance would be, and usage would be, pollutants that have a greater concentration along heavily trafficked roadways. And that would be NO2 and to some extent fine particles. Elemental carbon would be another such pollutant, and pHs, ambient pHs, when there's not a lot of wood burning going on, would also be mostly related to traffic emissions. And then it was an interesting study that found an association with traffic-related air pollution and insulin resistance. So potential mechanism, air pollution can induce oxidative stress, and that can lead to local inflammation in the airways, which can then spill over into the systemic circulation and cause systemic inflammation. I've shown that in some of my work with regard to ozone exposure. PM2.5, fine particulate, has in an animal model induced adipose tissue inflammation, like we see in adult obese humans, and insulin resistance in a model of diet-induced obesity. This hasn't been directly replicated in humans, but it's pretty strong evidence of a possible mechanism. An obese mouse. So, it's an hypothesis of mine, and I'm not the only one. Rob McConnell of the Children's Health Study, who also has one of the Children's Environmental Health Centers, also subscribes to this hypothesis that exposure to air pollution in utero and early childhood increases the risk of abnormal glucose metabolism later in childhood and risk of obesity later in childhood. These are the overarching hypotheses here. So I'm going to now tell you about the study that we're conducting in Fresno as part of our Children's Environmental Health Center. And our aim one was to determine whether chronic exposure to ambient air pollution, but especially pHs, because my colleague Catherine Hammond at UC Berkeley is particularly expert at measuring pHs, and we have a very nice pH exposure model where we can, based on extensive monitoring data, where we can assign pH exposures to any residence, any location in Fresno. And so our hypothesis or aim was to show whether chronic exposure to air pollution increased hemoglobin A1C. So rather than trying to diagnose diabetes in these kids, we're looking for a, if you will, preclinical marker, hemoglobin A1C. For those of you who don't know, glycosylated hemoglobin, that's hemoglobin A1C, is a direct reflection of glucose metabolism in the subject, and increased BMI, and increased levels of a marker of oxidative stress, 8-isoprostane, which is a product of lipid peroxidation that we have validated with regard to an epidemiologic study with my colleague Nina Holland at UC Berkeley, and we've also validated it in a controlled human exposure study in my lab at San Francisco General, where we expose people to ozone. C-reactive protein, a biomarker of systemic inflammation. The adipokines that Dr. Lamero and I have both mentioned, leptin and adiponectin. And then a marker of lipid metabolism, high-density lipoprotein. Now, for those of you who get your lipids checked, high-density lipoprotein is the good type of cholesterol. A higher level is promoted by healthy lifestyle. Why are we getting it here? Well, we're trying to avoid getting fasting blood levels. And HDL does correlate with metabolic syndrome fairly well, and you don't have to get it with a fasting specimen. So I want to talk about our specific aims 2 and 3, because here we're talking about mechanism. And I alluded to this again with my comment to Dr. Lamero. We also hypothesized that chronic exposure to air pollution induced T regulatory cell and T effector cell, these are lymphocyte populations, that we've already shown this immune dysregulation of these cells, especially the Treg cells in our Fresno kids. And so we want to determine whether that sort of known effect is associated with hemoglobin A1C and increased BMI. So just a little bit of immunology 101 here. So T lymphocytes are important cells in the immune system. T effector cells are the cells that you probably heard a lot about with regard to the hygiene hypothesis, which I suspect many of you know about, which suggests that because we have decreased exposure to various bacteria and other and parasites in our environment compared to the way we were initially living before modern civilization, there's been a shift from a predominant Th1 phenotype of lymphocytic cells to an allergic, which Th1 fighting infection, Th2 allergy, and the T regulatory cells are sort of above the effector cells in the immune control system. And T regulatory cells can suppress the immune response, either Th1 or Th2. And if T regulatory cells are dysfunctional, you may have problems with your immune response. And we have shown in Fresno that there's suppressed T regulatory function related to air pollution exposure, specifically PAH exposure. And with our colleague Kari Nadeau at Stanford, we've even come up with an epigenetic mechanism by how this might occur, that the FOXP3 gene, very important for T regulatory function, is hypermethylated with exposure to PAH, and that suppresses T regulatory function. And we showed that was associated with both increased asthma severity in a cohort of asthmatics, but also increased risk of allergies as well. I'm going to show the FOXP3 gene with CpG islands that are methylated. So we have an epigenetic mechanism for this immune response. And so we know that it's associated with asthma and allergic disease. The question is it associated with obesity and diabetes? And it's purely speculative. We don't have any data to support this, but I was pleased that our reviewers for our Children's Environmental Health Center proposal thought it was a reasonable hypothesis to test. So we're in the process of doing that. Our study design is to follow several different age strata of kids. We have a birth cohort, a child cohort, and an adolescent cohort. And for all participants, we're getting this list of various biomarkers and clinical tests, if you will. And then we have estimated air pollution exposure from the prenatal period up to whenever we see the kids. And I'm going to I don't have any data to show you in terms of testing the hypothesis. It's too early, though Nina Holland has just finished our Adolescent results for the first 100 adolescent kids, but I don't actually have them yet to show you. But these adolescent kids are representative of the Fresno population. That's pretty striking. At least the Fresno population that we were able to recruit. As you might expect, there's a high prevalence or proportion of Latino kids. But look at the percent of those with incomes below 15,000. Even in Fresno, you can't live on that. So we have and also look at the prevalence of asthma in our cohort, 35%. This, part of the reason we're studying air pollution in Fresno is because of the high asthma rate there. So just distribution of some of the anthropometric and hemoglobin A1C data. Again, this is sort of very crude. Well, actually, the data aren't crude, but we haven't associated them with anything yet. This is just the demographic or the anthropometric data. You can see that the mean BMI is already overweight, and the median is even, and in terms of body fat, also high. And hemoglobin A1C, when you compare to NHANES normal values, my table above doesn't even have it split by sex, but you can see if you look that the kids are overweight and the hemoglobin A1C is higher than we would expect. Kids are fatter and have a little bit less good glucose metabolism. So next time I'm asked to speak about this, hopefully I'll have some data about adipokines, and we're soon going to be associating the air pollution variables with the outcome variables. So in summary, the prevalence of both obesity and diabetes is high among Latino youth in the San Joaquin Valley. Air pollution may increase the risk of both conditions by inducing oxidative stress and systemic inflammation. We think that pH-induced immune dysfunction may play a role in this pathway, yet to be proven. And our study design allows investigation of this potential pathway in multiple age cohorts. And so I just close by acknowledging my colleagues at both Berkeley and Stanford, as well as UCF Fresno, and our collaborator Fred Lerman from Sonoma Technology. Jennifer's in the audience. And wanted to thank our funders, EPA and IHS. Oh yes, we're also collaborating with Cal State Fresno. I shouldn't minimize that. And with that, I thank you.